just a bit of background about me. So as you might guess from my accent, I'm not from the UK, and I'm actually from Italy, from Sicily, where I did do my bachelor and master degree in applied mathematics. And after that, I thought that I wanted to do something with my knowledge in maths. And that's why I moved to the UK, where I did a PhD in computational biology in Cambridge. And that gave me the opportunity to apply my mathematical, statistical, and computational skills around cancer research. After PhD, I thought I really enjoyed teaching. So I thought, okay, why not changing career? So I went for 3 years into a teaching career in mathematics, which took me to the north, where I then stayed from 2019 when I joined Teesside University, where I'm now associate professor, and I lead, can't move that, but I lead the AI group there. So what I'm going to talk to you about today is about the research that I've been doing over the last 4 years or so. And as you might have seen from the title, it's all around AI for cancer biology. But towards the end, I also added a few slides on the projects that we do around biotechnology as well. So if you work with AI around different themes or scenarios, you might all be familiar with these, which is just normal neural networks, which is what it's nowadays used for any AI approach. Of course, many more methods, more complex methods are developed starting from these. What we do is through these complex methods, we try to integrate what we call multimodal data. So we're not just putting only one data type in here, which could be transcriptomics or clinical data, but we work with data that gives us an overview of how the patient is progressing or how the single cells are changing. So in this case, we can have transcriptomics, clinical, proteomics, or even images all together. We'll feed all this information within an intelligence model, which is most of the case based on neural networks and complex integration of neural networks. And then we try to deliver some outputs. These outputs can be different. We could predict the survival of the patients, or we could try to understand if that tissue was a tumor tissue or was just a healthy tissue, or we want to do some prediction in terms of drug response. So considering what's been happening in the past, or considering, for example, the metabolic network of that specific cell type, we want to predict if that specific cell will respond or not to a specific drug. Now, all this is very exciting, and that's what I'm really passionate about. But there are a few problems around what's actually happening in the middle of these methods. Now, we are integrating a bit of multimodal. I'll try to do this full screen again, just to see if that we can see what's at the bottom. If not, we'll have to live with this. So there are a few problems which are about, we are integrating different multimodal data. So it's like we concatenate lots of numbers. We can translate images into numbers as well. But then we are throwing everything together into the same network. So there is no real biological knowledge in here because we're just using data-driven model. They're trying to give a connection, just to create a connection between some data that's in the input and some numbers that's in the output. Those models that we are developing there, they do not know anything about biology. That's when we started thinking about how we can improve these models so that actually the prediction that we get is related to the biology or has some kind of biological meaning. And that's what took us to start exploring metabolism. Now, we know that any change that happens at the phenotypic level, if it's, for example, cancer cells, is a normal cell, or how cell behaves, are all connected to what happens within a cell in terms of their metabolic changes. Now, when we talk about metabolism, we talk about the set of chemical reactions that allow the cells to survive, to grow, and proliferate. And usually there's sets of reactions are connected together into a larger networks that are called pathways. And we tend to work most of the time with these metabolic pathways, which again are a set of chemical connections, a set of chemical reactions. This is very important because by looking at these biochemical reactions and how these chemical reactions are changing across different cell types, we can understand why a cell is behaving in one way rather than another way. And this is all connected to their metabolic network. Now, the good thing is that as a mathematician, I was really interested in translating this biological knowledge into some kind of mathematical representation so that I could have integrated these models within that AI approach. And this is the reason why we moved to what's called genome scale metabolic modeling, where we can represent these reactions into a matrix. So something that's can be mathematically represented. So if we think about reaction, it's just a metabolite in, so something that comes in, and a metabolite that comes out. So if we think about this single reaction, we can of course represent this as a in a matrix as that reaction is consuming, so that -1 indicates us that we're consuming something and we are producing something else. Those somethings, those symbols are actually molecules. That can be glucose that's changed into pyruvate or lactate that's being produced as part of a specific reaction. So we are translating all these reactions, these metabolic reactions, into something that can be interpreted into a mathematical way. And this is not something that we invented. This has been around for many years. And in this nice review paper from 2019, I'm happy to circulate the slides. There is the review title at the bottom of the slides, but they show how these type of models are developed, and they're all based on some genome knowledge. So there are models, these metabolic models are available for different species, and nowadays for different cell lines as well. So we're talking about before about E. coli, but also about different cell lines or different subtypes if we talk about breast cancer. And these models usually come as simple models, or you can add, if by doing some research, you have discovered that specific cancer type behaves in a different way compared to what's in the model, you can manually add extra reactions. And then these reactions are converted again into that matrix I was telling you about. And through optimization process, you will get what's called flux vector. So the flux vector you can think of as a number that tells you how fast each one of those reactions is happening, is taking place. And of course, if you then compare these flux vectors of different species or different cell types, you can understand which reactions are more active, let's say, in one cell compared to another, and understanding why that's happening. Is it because there is abundance of a specific enzyme, or does this depend on the medium where that specific cell line has been grown? So this will be the key factor that will tell us how these metabolites are being consumed or produced. We have recently developed a general framework on how you can integrate this not just on bulk RNA-seq data, but also on single cell. And the process is more or less the same, either if you go top or the bottom. There are usually some predefined methods or models that you can use to develop. In this case, we were looking at tissue-specific models, but this can be species-specific models, and then add some extra knowledge from external database, and we will get down to this matrix, which is what we're going to focus on, which is a matrix that tells you the flux, so it's the number for each reactions that you have in the model, will tell you how fast that reaction is taking place for all the samples that you have. So this is what we are interested in this type of matrix. And of course, there are methods that can generate these if we talk about bulk RNA sequence, but nowadays, in the last few years, there are lots of methods have been developed also for single cell. And as you can imagine, the single cell are a bit more accurate because they can tell you exactly what's happening at the metabolic level between an immune cell and the stroma cells, so between B cells and the T cells. Okay, so on one side, we have the AI approaches. On the other side, we have the metabolic modeling. They have pros and cons on both sides. And what we do in our group, we merge both models. This is again part of the framework that we developed. It's in there is the link at the bottom if you want to look at, but it's in Trends in Biology. And we start with the data. So as I was mentioning before, we have lots of different data types that we can integrate. We have the metabolic knowledge, but we can also integrate some knowledge specific related to the drug that we want to use. If we then work with images, we use some feature extraction methods to extract key features from those images. And then we move to the usual models. Now we're not really focusing on neural networks anymore. Usually we are now focusing on something that's kind of combination of, or more complex combination of neural networks, which are the attention-based models or transformers. And we can integrate the data across different three scenarios. Some of you may be familiar with this. Early integration means that once we have all those numbers at the beginning, transcriptomics, metabolomic, and so on, we just push everything together into one single vector, and we feed this into the model. When we talk about intermediate integration, we extract latent features from each one of those modalities, and then we'll integrate them at the end. Or late integration, as you can imagine, you just run the model for each one of those modalities, and then you just integrate the final prediction. This is usually based on probability distributions or majority voting approaches. And then the final task is always the same. We are trying to, predict classification, want to do some classification tasks or predict drug response. But the key things here is about the bottom panel here. So the bottom panel tells us things that we couldn't have discovered unless we use some unless we integrate some biological knowledge. And this is what I'm going to focus on in the next few slides. So we can have a mechanistic understanding of what's happening within the cells. We can identify important features. This is done through explainability analysis, but we can also look at the H&E images and try to understand what happens in terms of tumor microenvironment. Okay, so approximately in the next 10 minutes, I will go through some cancer-related applications and how we are doing this. Now, this story started actually in 2019 when I got the first grant to actually apply this on breast cancer patients. So the idea was quite simple at the time. They asked us, or they gave us some funding to explore this initial idea of integrating different data types. At the time, we were looking at genomics, we were mostly looking at transcriptomics, fluxomic data, and clinical data, using those genome scale metabolic models to create the metabolic profiles for each one of those patients, and then using simple machine learning and survival analysis to make a prediction on patient survival. So it was just a survival prediction model that was using three data modalities: transcriptomics, fluxomic, and clinical data. And the outcome here was just trying to understand why there were differences across those patients and how we could relate those at the metabolic level. But that project was actually stopping at the prediction. We were doing the prediction, and that was it. So then we thought, okay, how can we push this one step further rather than just doing the prediction? And that's when we started embedding explainability approaches as well as lab experiment. And this is another paper that's currently under review where we are starting from transcriptomics, embedding those transcriptomics into the genome scale metabolic modeling. This is about breast cancer, and we are generating those fluxomic. Then we do the usual data pre-processing, feature selection. We applied, in this case, very simple machine learning models, and the outcome, or what we're trying to do, was more or less the same, comparing models. We were doing survival analysis, pathway enrichment analysis, but this bit at the bottom was what we were most interested in. We wanted actually to investigate why the model was making those predictions and what actions we could have taken on those insights. This is what we got actually when we ran SHAP analysis. For those of you who are not familiar with SHAP, it's just a kind of a quick test that you can run on your model to check which features are having an impact on your prediction. So in this case, we were integrating clinical fluxomics and transcriptomics, and we were asking, okay, tell me which features are important. And we were quite surprised in seeing that actually the clinical feature, well, this is not very surprising, but the clinical features like the age or which cohort they belong to was identified at the top from by most of the methods. But after the clinical features, then came the transcriptomic features, which are the ones that starts with T, then there is the G number, and the fluxomic features. So there was a mixture. If we can see here, there is a mixture of C and F, C and F. Here, this method was giving higher importance to the fluxomic features. This was a mixture again. So we start thinking, okay, so it's not true what has been seen so far, that only the clinical features and the transcriptomic features are having a role when making prediction, but actually fluxomics can also tell us something extra. And then we started doing lots of analysis on these. I'll probably leave this, I will not get into detail, but again, happy to circulate the slides afterwards. But we identify some genes. For example, here at the top, we have some important genes for breast cancer. And two of these were known to be involved in cancer progression. So that was something that we knew already. But what's important about these methods is that then we can also identify genes that are not known yet for that specific cancer type or for stratifying subtypes. And this allowed us to select a few key genes that are now being used as a validation. So we are validating these across different breast cancer cell lines, subtypes, cell lines. And when we do a simple UMAP, we can actually see that those transcriptomic features are relevant. You can see here that just by using the transcriptomics features identified by the SHAP analysis, the top 500 we selected here, those subtypes, those breast cancer subtypes are being very well distributed. Again, we can go one level down and see how these genes are impacting us. But if we look, for example, at the PCIB plot, we can actually see which genes are having the highest contribution in identifying the difference between those subtypes. And this is quite important because, as we know, different subtypes will need different treatment, or they are responding differently to the same drug. And identifying which genes are having the highest importance or the highest contribution in separating them, it's actually very important to understand drug development or targeted therapies. This was done just in the transcriptomics. By integrating the metabolic knowledge, we could do the same just on what was happening in the metabolism of those patients in this case. And again, I'm just leaving this on the slides. It's just to show you that we can identify metabolic changes that are helping us in understanding why there is a difference between these breast cancer subtypes. And it wasn't surprising that most of them were related to immune response. What else we can do? We can get the usual KEGG pathway analysis, enrichment analysis through the metabolic pathways. But the things on the right is something extra that we can do. The thing on the right is just a pathway. In this case, we're looking at glycolysis with CCA and the fatty acid oxidation. And we can see where differences across patients are actually taking place. The red color is showing you the highest difference. This was a log 2-fold change in high-risk patients. So the red colors is actually where we can spot differences. And this is not about transcriptomics. This is exactly where in the metabolism things are actually changing. And we're now doing this at the single cell level. So we're validating single cell level and translating this at the spatial transcriptomic level, and with a closer look at the tumor microenvironment. So this was the first part of the analysis. But remember, we did this just on bulk data because it was the TCGA sample, TCGA data set. So it's a public available data set of bulk RNA data. So we thought, why don't we take this one step further and we apply the same technique at the single cell level? Because if we apply this at the single cell level, we can actually see where the differences are happening. Because at the moment with bulk, you can only see the differences between patients, but within that transcriptomics, you're taking a mixture of cell types. It's just coming from the biopsy, but it's a mixture of cell types. If we can translate these into single cell, so just looking at a specific cell, we will be able to identify metabolic changes across cell types. And this is what took us to collaboration with Weill Cornell Medicine. This is actually part of an EPSRC grant that started in February. And we're kind of replicating what I've been telling you about, but this time at the single cell level. Still for classifying breast cancer subtypes, but we wanted to do this, to understand this across different cell lines. And the cell lines we were looking at were related to 5 different breast cancer subtypes. This was just the initial framework. We are still integrating different data types across a multimodal machine learning approach to do the biomarker identification as well as explainability analysis. And in this case, we didn't use TCGA, but we focused on a paper on the data provided by a research center in Italy, where they identified more than 35,000 cells across 32 breast cancer cell lines, which then were split into 6 breast cancer subtypes. So the idea was using these transcriptomic profiles, develop the metabolic models for those individual cells, and then trying to understand the difference using machine learning and AI approaches. And this helped us in identifying changes at the reaction level. So we could see across subtypes, those were the subtypes in that paper, and these are the fluxes that we generated. We could see how the glucose consumption, in this case is negative because it's being consumed, is changing across subtype, how lactate production is connected to that, and simultaneously we can do this across different metabolites. So the metabolites are those molecules that have been consumed or produced by reactions. So we can analyze the changes at the metabolites level, but we can also see exactly where those changes are happening in the huge metabolic network. So we know there is more glucose consumed in a specific cell, and that, of course, will be part mostly of the glycolysis. But what about all the other metabolites? You can see exactly at which metabolic reactions or where in the metabolic network these changes are happening. And of course, by looking at these reactions across cell types and breast cancer subtypes, you can get, even by doing a simple differential expression analysis, you can realize what's going differently across breast cancer subtypes in this case. And we're now at the process of validating the things that were not known in the literature. So we identified some reactions that were behaving differently across cell types, across cell lines. And we're now validating these in collaboration with the Valencia Biomedical Research Foundation. Okay, so that was the second part. We started from bulk and we just used single cell for validation. Then we used single cell for actually implement the entire pipeline, and that allowed us to look at differences between cell types. But then we thought, okay, why don't we apply this into something a bit more concrete, something that could really help clinicians or help, in this case, breast cancer research. And that's what took us to collaboration around immunotherapy. So the idea is still the same, applying this framework I've been telling you about to understand what's happening across, in this case was mice, that were responding or not to immunotherapy treatment. This is done with another group, still at Weill Cornell Medicine, led by the first author of this paper, Roberta. And what they did with these mice, they had two groups. One was receiving anti-CTLA-4 treatment. The other one was just control. And anti-CTLA-4 treatment is type of treatment is similar to PD-L1 blockade, if you're familiar with that, but stops one of the connection, let's say, that's on the T cells, and avoid that the T cells becoming silent. So by using this type of treatment, we are not stopping the T cells from doing their job. T cells is the immune cells. So by blocking that particle on the T cells, we are allowing actually T cells in doing their job, and we want to see how this process is happening or how this is taking place into we looked mostly at T cells in this case. This is what we get. So it's like a network, still the same metabolic network we were looking at. It's probably arranged in a different way. But the interesting thing was that they also had metabolomics data. So we tried to correlate these with the metabolomics data, and we were able to identify where changes, this is just glycolysis, where changes were happening in the glycolysis pathway. And this was important because by looking at changes in the glycolysis pathway, you can understand which T cells or which B cells are getting activated or are actually doing their job as immune cells. We then looked at also some single cell validation across different treatments. In this case, we looked at anti-PD-L1 treatment. So we checked across different 4 pathways. They're here on the right. We checked what was happening at the metabolic level and why we could see differences between those patients that were responding or not responding to one of these 2 treatments. And I'll leave this on the slide, but it's just to tell you what we're actually looking at the moment. We are trying to understand at the metabolic level why B cells are behaving in a specific way or in another, so going in one direction or in another direction, depending if they have an anti-PD-L1, so immune cell point blockade, or just chemotherapy. And the good thing is that we had the data for different treatments. So we had the responders before treatment and post-treatment, but also the people that did not respond before and after treatment. And this helped us, or is helping actually us a lot in understanding where those differences are happening. And I want to finish this cancer side just by quickly running through other applications. So at the moment, we're not integrating images, but in the examples that, or in the projects I've been telling you about, but we'll also do lots of multimodal integration with images. And in lung cancer, for example, we talk about radiological images. This was a recent work where we actually had the challenge, which is probably one of the most common challenge for those of us working in multimodal, of having low samples with all the data modalities. So in this case, we were looking at images, so CT scan images, RNA, which was a transcriptomic, clinical data, and some biological information. And we had, across these 3 different modalities, we had only 130 samples, which it's not a lot if you want to do some deep learning model. But we managed to do this through some applying this more advanced cross-validation approaches. And at the end, yeah, we will always looking at the usual performance of the model, but also some explainability. And the good thing of this model, this work, was that we involved some radiologists to actually check what the model was doing, or if what the model was predicting was good or not. And we were quite surprised. We were quite happy, actually, not surprised, happy to see that actually the model was performing well. And we went one step further and say, okay, rather than just doing clinical transcriptomic and radiological images, why don't we add other omics as well? And this is a paper that's currently completing, will probably be submitted soon. In this case, it was about ovarian cancer. We integrated images, gene expression, and fluxomic data, and we still do the same thing about, in this case, where we're using early integration for survival prediction. And yeah, just don't want to spend too long on this, but it's just to show that when we actually integrate the three data types, we're getting the highest performance in terms of model accuracy. But as we know, it's not all about performance. We also want to do some validation, and by doing the validation, we can actually identify again where things are changing and understanding why those things are changing. I'll leave this on the screen again. I'm not going to go through all these findings, but this is actually what's helping us in identifying where which genes are known or which changes are known, in this case in ovarian cancer, and which ones are not known yet. And that could be validated then externally. And we are in the process of doing that in order to move forward the research in this case. And we're also involved in a few clinical applications. This is in collaboration with as part of a grant with the British Society of Breast Radiology and the NHS, where they, and specifically is the Foundation Trust Gateshead, and they had lots of data across different modalities, but they also know what response patients had. This was chemotherapy. So the idea of this grant that's currently running is to try to develop a model that could connect those data with the response that the patients had in order to understand what's happening, why some patients are responding to that treatment and some patients are not responding. They ideally would like to find 2 or 3 of these modalities that are actually really important in driving that prediction, so that we can work with consultant radiologists to better identify patients for targeted treatments. And we do a lot of work on other modalities as well, so specifically around infrared spectroscopy. This is another grant, an EPSRC grant through the ClearPath AI network. And the idea is merging 2 modalities. In this case was the H&E images with infrared spectroscopy to predict if that tissue sample was malignant or benign. And the final idea will be to identify key regions in the H&E spectra, and so in the H&E image that could connect with specific peaks on the spectra, so that at a later stage we can just only rely on spectra rather than waiting for H&E slides. And finally, just the last 3 slides of this talk about the biotechnology applications, and most of these grants have been supported by VBSRC. So the first one is about the metabolic modeling of metallogenic archaea. So archaea is bacteria that generates methane under specific condition. So what we did, this was a collaboration with Prajeta company in Austria. We applied, or we used the data that was already available around the metabolic network of these bacteria, and then we manually changed a few things to develop the final genome scale metabolic modeling, and then we applied, in this case, just FBA and FDA to understand how those strains will behave under different conditions, and we looked specifically at CO2 and oxygen, different levels of CO2 and oxygen. And then we identified which fluxomic or which reactions and pathways were actually more involved in this and could inform the company, in this case Crogete, on how to proceed for improving their manufacturing process. A similar application was done with Fujifilm. This was, again, a BBSRC project through the Metal Biology Network. And we looked, it was a similar idea, but we looked at E. coli and how E. coli would respond across different experimental bioreactor data. So they've done some different experiments with different pH and temperature, so different conditions. And they wanted to correlate this with the growth rate of the bacteria, the E. coli in this case. And again, we applied machine learning to correlate input and output in this case. And finally, another project with Porn. This time was in collaboration through the National Biofilm Innovation Center. This was mostly based on images, and we developed a model that called automatically identifying spot biofilms from images that were coming from the microscope. So the engineer was taking a sample from fermenter, generating the image, and then the model was able to automatically detect those biofilms, informing when the fermentation process should stop or not, because based on this, on the quantity of this biofilm, then the broth is not of a good quality anymore. So we support them in the development of this as well. And I'd like to finish with some self-advertising. If you also work around on multimodal AI, especially in bioimaging, I'm one of the guest editors of this collection around AI for multimodal biomedical imaging. If you're interested or if you want more information, feel free to either check the website or contact me. And I'm also part of the, I'm actually one of the co-organizer of the AI for Women Health interest group at the Alan Turing Institute. We do organize monthly meetings. We had one just last week. And if you are interested in this field, feel free to connect with us. Drop me an email if you want to be included in the next meeting, or if you want to present your research as well. And what's next? As we know, spatial omics is a big thing now, and that's what we hope we can contribute to with the method that we're developing in the model in the group at the moment. And with this, I'd like to thank the funders and thank you all for your attention, for the invitation again. Thank you.